And so our first talk is going to be given by Brittany Borbong, Michelle Cruz, and Fang Yung Hong, and they're going to talk about a meta-analysis of coastal populations, genetic diversity throughout their range.
So like Suzanne said, our presentation is a meta-analysis of coastal populations, genetic diversity throughout their range. So in population genetics, there is a widespread assumption called the abundant center hypothesis that states that within a species' natural distribution range, at the center of the range, the populations are more abundant, and as you get closer to the edge, the abundance declines. So in this figure, a species' natural distribution range has been generated into an image, and a center has been calculated. This red spot around it is showing a high level of abundance, and as you get closer to an edge, the color starts to lessen, indicating that the abundance of the species population is declining. Now, despite the fact that this is a widespread assumption and highly used in population genetics, this hypothesis has rarely been rigorously tested. And among the studies that have rigorously tested it, very few support it. And so in 2008, Eckert et al. Published a paper in which they looked more into the abundant center hypothesis. So in 2008, Eckert et al. Published the paper Genetic Variation Across Species Geographical Ranges, the Central Marginal Hypothesis and Beyond. And the central marginal hypothesis is just another name for the abundant center hypothesis. So Eckert et al. Hypothesized that the genetic diversity among plants and animals would decline as populations reach the edge of their range. And so their hypothesis was that the abundant center hypothesis was accurate. They used a vote counting method, which is simply a method in which you categorize the studies you're looking at into one category that says a study does support your hypothesis, and another that says the study does not support your hypothesis. And so they used a vote counting method to analyze 134 studies that looked at 115 species. At the end of their study, they concluded that 64% of the studies analyzed did in fact support their hypothesis. However, because they used a vote counting method, they were unable to account for variability between studies, and they also could not place weights among studies. So a study that only used 50 samples was weighted the same as a study that used 500. And another downfall of a vote counting method is that it is very subjective. It is up to the reviewer to decide if a study does in fact support or refute the hypothesis. So it's not a 100% objective method. So using Eckert et al. As our inspiration, we came up with our own question that we wanted to study. And so our question was asking if within coastal species, if genetic diversity is affected by the species location within their own natural range. And the reason why we decided to study coastal species is because in 2006, Sagren et al. Published a paper in which they looked into the abundant center hypothesis, and they mentioned that coastal species are really interesting to look at because of their unique geometric ranges. The species tend to cover less environmental variability, and they also tend to have fewer confounding factors because they are usually one-dimensional and along a coastline. And so using Sagren and Eckert, we came up with our own hypothesis and we hypothesized that coastal populations' distance to the center of the range is positively correlated with genetic diversity. So in contrast with the abundant center hypothesis, we hypothesized that coastal populations' genetic diversity would increase as you got closer to the edge. And the reason why looking at coastal species is important is because coastal species are heavily endangered and threatened, and they're heavily affected by environmental factors such as climate change. And to have better conservation efforts placed on these species, we should look at the individual populations as opposed to the entire species as a whole, because different populations could be affected in different ways by environmental factors. So if we were to conclude that different populations were genetically diverse from others within the same species, people would be able to better specify conservation efforts to specific populations as opposed to one method for one species. Also, another issue is that for species to be put onto a watch list for endangered and threatened species, they need to show a level of genetic differentiation between other species. And since coastal species aren't studied as heavily as other ones, they are kind of assumed to have a high level of homogeny, and therefore they aren't placed on a lot of watch lists. So if we study the genetic diversity between populations within the species, hopefully in showing that they are diverse from one another, they will be able to be put on watch lists and receive the conservation efforts that they need. So in our study, we used a meta-analysis, and a meta-analysis is a statistical technique that is used to compare the results of similar studies. And we decided to use meta-analysis because it is more rigorous than a vote counting method because it uses an effect size to compare among studies as opposed to a subjective categorical method as the vote counting method is. A downfall to meta-analysis is that you do a literature search and you use those literature searches within your meta-analysis. However, there could be papers out there or studies out there conducted that had insignificant results and were therefore not published. And so it's difficult to find those studies and include them in your meta-analysis, so they tend to not be. And it's difficult to know if those results will impact your study. So if those insignificant results will actually have a significant result or significant impact on what you're looking at. And so one way to correct that downfall is to calculate a publication bias in which you can calculate a number of unpublished studies that would affect the results of your meta-analysis. And so for our meta-analysis, we decided to only focus on 6 taxa. We decided to focus on sharks, fish, plants, mollusks, corals, and reptiles. We decided to focus only on these 6 taxa because they are heavily documented online and the information was easily accessible. To measure genetic diversity, we decided to only use studies that had nuclear microsatellites because they are the most used nuclear or genetic marker in the past 20 years, and they are more accurate than allozymes and SNPs. And to measure genetic diversity, we looked at expected and observed heterozygosity, alleles per locus, allelic richness, inbreeding coefficient, and genetic differentiation.
So in deciding if we could include or exclude papers in our meta-analysis, we use the following criteria. We were looking for studies that focus on coastal species, that use microsatellites as their genetic marker, and studies that have appropriate sample size. So studies that at least had 4 populations with 10 individuals per population. Studies that looked at species that had documented sample sites. So studies that either had coordinates or maps showing where they got their samples. And studies that were looking into species that had documented range maps. So we wanted to focus on species that have documented range maps because without a range map, it's impossible to define a range or a center. And we obtained these range maps on online databases such as Aquamaps, Map of Life, or the UCN Red List. Also, papers that include genetic measures, like Brittany mentioned earlier. So we're looking at heterozygosity, allelic richness, number of alleles, alleles per locus, and FIS and FST. Also, papers that were looking at non-invasive freshwater migratory, or that did not collect their samples from hatcheries. So we conducted a primary literature search on Web of Science. Our first search was a combination of the top combination of those keywords, and that resulted in 574 papers. We then refined our search again, and that gave us 42 papers. So obviously 42 is far less than what we expected or wanted. So then we expanded our search again by adding allelic diversity, not freshwater, not migratory, non-invasive, and this resulted in 156 papers. After reading those papers, we had to exclude 142 papers because it did not meet our criteria, so that left us with 14 papers. We then had an additional 21 papers that we were able to obtain by including common name of higher taxa into our search. And out of those 21 papers, we had to exclude 17. We then had 3 additional papers that we obtained from a colleague of Sean. And out of those 3 papers, we had to exclude 1. We then added reptiles to our search, and that gave us 3 additional papers. Unfortunately, we had to exclude those same 3. At the end of our literature search, we had a total of 21 papers that met our criteria that we could used in our meta-analysis, and that covered a range of 19 species. After looking at the range maps of those 19 species, we categorized the range maps to 2 categories. We had one-dimensional range maps and two-dimensional range maps. Now, one-dimensional range maps, they span along a single coast, and their width is less than 10% of its total length. Two-dimensional range maps, they can span along multiple coasts and oceans, and their width is greater than 10% of its total length. Defining a center. Now, for one-dimensional range maps, we define the center as the midpoint of the addition of linear segments along the coast. So for one-dimensional ranges, we did take into account the curves of the coastline. We then measured the distance from the center to the sample sites using the same method of delineation. For a two-dimensional range, we used ArcGIS to find the mean center of the range map. We then measured the distance from the center to sample sites by point to point. So for two-dimensional range maps, we did not take into account the curves of coastlines. In cases that we could not find a range map online, but were able to find point maps, we used ArcGIS to create a convex hull that encompassed all of the points, and that created a range. We then treated the point maps as a two-dimensional range map. So we used ArcGIS to find the mean center, and we measured the distance from the center to the sample sites point to point.
For the data analysis, I'll walk you through some of the major steps and ideas behind our models. So the most fundamental idea is displayed here. This is a linear correlation graph between alleles per locus and a relative distance from the center of the range of Pagras major to each of the sample point. The R value, or the correlation coefficient, measures the linear correlation between two variables, and the response variable must be continuous. And we told R to calculate the R values for each pair of genetic variables and distance for each species. And in this graph, we see that as relative distance from center increases, alleles per locus increases. But this is somewhat dependent on this lonely point here in the corner. Which means that the more samples we have, and as well as broader and the more even the distribution of the samples is throughout the range, the more we can trust the R value. Well, what does this tell us? We have to put greater weight on studies that have better sampling methods or more biologically stable populations, which also means that we have to use factors to explain the variability of R values for different studies. Before jumping into analysis, we did an independence test to validate our choice of genetic variables from a statistical perspective. And here we see that allelic richness is highly correlated with many of the other genetic variables. So that's why we eliminate it and focus on the other 5. So we first, these are the methodological and biological factors that we consider important. Michelle has already covered on the point versus range map source, and range dimension. And here I would like to introduce some of the more interesting metrics that can either qualify or quantify how the sample how is the sample cover the whole range. So see, for relative range coverage of samples, we divide the area of the sample convex hull by that of the total range, so that we have an idea of how far or how big the samples cover the whole range. And another metric is relative distribution, which takes into account all the distance from the center to sample points. And we calculate it by taking the difference between the maximum relative distance and the minimum, so like this line here. And another one is relative direction, which considers which direction the samples were taken relative to the range center. And so these are the results of single factor test. We see that only one pair shows significant correlation, and the other either shows either marginally significant or insignificant at all. We apparently would like to know more about what's going on in our models, so that's where we came up with our multi-factor models. We choose 6 groups of predictive and response variables from factors that were significant or marginally significant. And this way, we were able to avoid the complexities of non-independence of data from 2 variables, and also partition out some of the important methodological factors to focus on more important biological factors. And the results show that 5 groups have significant p-values, except for the last one. Here's one case that we found particularly interesting and easy to understand. The 2 boxes for 1-dimensional range and 2-dimensional range did not overlap 0, which means that which suggests there is this association. And looking at the p-values, it turns out that 1-dimensional range has a significant p-value, but not the 2-dimensional. And which means that the signal for 1-dimensional is stronger than 2-dimensional. And we see it here as distance from center increases, the genetic diversity increases for 1-dimensional. That support our hypothesis. We can explain these differentiation between 1 and 2 by thinking that 1-dimensional ranges usually have less environmental variability, so probably will give better genetic signals. And so these are all the 5 groups that have significant correlations. And another group that I would like to mention is here, expected heterozygosity versus taxa. And the results here show that as the distance from the center increases, the heterozygosity of plants decrease, which contradicts our hypothesis. We thought about some explanations. For example, it might be that plants don't move, populations at the edge may have less interaction than population in the center. And also because a limited dispersal by pollen and seed may result in a decrease in the heterozygosity further away from the center. And also, plants will usually grow in an optimal condition and disperse from there. So the soil conditions and climate conditions may be different further away from the center, and thus causing the decrease in heterozygosity. So to conclude, our meta-analysis partly support a hypothesis. However, in order to use geographical distribution as an explanatory factor of genetic diversity with confidence, we have to look at different taxa, range dimension, the sample distribution, and so on. So in other words, the complexity of this study prevents us from making any assertion or generality. So in the future, we would like to include more studies, especially those that focus on different taxa, that take samples from the southern hemisphere or south of the center, and also use another genetic markers. So lastly, we would like to acknowledge the support from NIMBioS.
We'd like to thank the staff and faculty associated with NIMBioS, especially Eric Carr, who helped us tremendously by teaching us how to use ArcGIS. And obviously, we'd like to thank Kelly and Suzanne. Without their help and their support, we wouldn't have been as comfortable as we were and successful as we were during NIMBioS. And we'd like to thank the NSF for supporting and funding NIMBioS. And last but not least, we would like to thank our mentors who unfortunately could not be here. Sean and Julia were the greatest mentors that we could have. They supported us through everything, and were fantastic and helped us throughout this project. And without them, we wouldn't have been as successful and done as well as we did.